Just- Okay, we are live now. Thank you everyone for joining in today. We have Aniruddh with us, Aniruddh Maheshwari, and we have Ahan bhowmick with us. So both of them did a wonderful research project in the summer of 2024. And we also have Sharif, who is one of the advisors, and Muskan, who is also one of the advisors. So thank you all for joining in. It would be great to learn something wonderful. So I'll go to each of the kids and ask them to give a couple of lines of introduction. So let's start with Ahan. Ahan, go ahead.
Hello, sir. My name is Ahan Bhowmick. I'm from Washington. I'm in grade 9 and I study in Redmond High School.
That's great. And Anirudh, why don't you tell us a little bit about yourself?
Hi, my name is Aniruddh Maheshwari. I'm in grade 9. I go to Monte Vista High School in California.
Excellent. So the way this will go is that whoever wants to start presenting, you can start. And then when you want to go to the next slide, you just tell me, I want to go to the next slide. Or just say next slide. I'll just move the slide. Okay. All right. So please start.
Okay, next slide.
Okay, so the main parts of our project are the feature descriptors such as HOG, which stands for Histogram of Gradients, and LBP, which stands for Local Binary Patterns. Another part is the CNN architectures, and our model is based on the ResNet 50 architecture. Also, we have used data augmentation and stratified k-fold cross-validation.
Next slide.
Next slide. Anirudh can introduce the feature descriptors first.
So, feature descriptors are an algorithm which simplify images by capturing specific characteristics for efficient object search. So, what it does is that in an image, there's a lot of noise, and lot of noise, saturation, brightness, and many unnecessary factors. So, a feature descriptor algorithm keeps what's needed in the image and discards what's not. So, examples of feature descriptors are HOG, which we are using, and LBP, as well as SIFT, which stands for Scale Invariant Feature Transform. Next slide. So, we have decided to use the HOG feature descriptor because it's really good at finding edges and highlighting the sharpest gradients in an image. So over here, as you can see, there's a car in the foreground and a desert in the background. And after converting it using the hog feature algorithm, we can see that most of the unnecessary detail has been discarded and only the car and its edges are remaining. So we decided to use this feature descriptor because it's good at detecting brain tumors in brain MRIs where the brain tumor is small and isolated and its edges are visible and apparent. Next slide. How hog works is first we convert the image to grayscale, and when we do that, each pixel in the image is assigned an intensity from 0 to 255. So we calculate image gradients and angles to highlight these edges with the following formulas. Square root of gx squared plus gy squared, and for the angles, the arctan of gy by gx. So we apply this formula to the magnitude formula to every single pixel and its neighboring pixel to find all the gradients present in the image. And then we apply the this angle formula to find the orientation of this gradient. So now we have two one-to-one mappings of the image. So one is the magnitudes and one is the angle. So every pixel is given a magnitude value and an angle value. After this, we first divide the image into small cells. In our case, we've used 8 by 8 pixels. And we do something called orientation binning. So we have 9 bins, or essentially buckets, ranging labeled 0 degrees to 180 degrees. So for each pixel based on its angle value, we split the magnitude values to its nearest uh, closest angle values. For example, if we have a pixel with a magnitude of 100 and the angle value is 20, so based on the angle value and the magnitude value, we assign different numbers to each of the bins. After going through every single pixel, we add up all the values in the bins, and we construct a normalized histogram for this. We also do something called as block normalization, which essentially keeps it invariant to brightness and other factors. Next slide. In our testing, we noticed that the hog feature descriptor was not very accurate at detecting tumors where it was more gradual, where it was bigger, gradual, and more spread out because the gradients weren't as sharp or apparent. To combat this, we use the LBP feature descriptor, which captures local texture information by comparing each pixel with its neighbors. So this LBP stands for local binary patterns. So it finds the local textures present in the image. Next slide. So this works by first doing a neighborhood comparison. So we split the image into 3 by 3 grids, 3 by 3 cells throughout all the pixels, and every single pixel is compared with its 8 neighboring pixels. So if you have a 3 by 3 grid, we find the intensity of the middle pixel, and if any pixel in the neighborhood is less, is its intensity is lesser than that of the middle, then we give it a value of 0. And if it's more, we give it a value of 1. Finally, we concatenate all of these numbers and form a 8-bit binary number, which we convert to decimal and assign the center pixel that value. From this, we concatenate all of the results for every single grid and create a histogram of it. And over here, you can see that this image, after putting the LBP feature descriptor, we can see all the local textures present in the car and its background. Next. Finally, we did data augmentation. So data augmentation involves applying many transformations to the original image to firstly increase the diversity of the training dataset and to simulate real world variations. So over here we have an original image which is of a brain tumor in a brain, MRI, and we have done many data augmentations to it like zooming, distortion, shearing, and rotation. This is important because it improves the model's generalizations, and it increases the data set because of all of these transformations. It also simulates real world variations because MRIs can give distorted images or slightly rotated or flipped images, which makes the model um, better generalized for future unseen images. Next slide. So now that we've talked about all of these feature descriptors, let me hand it over to Ahan to explain the second part of our project.
Thank you, Aniruddh. The second part of our project is the neural networks. So, what are neural networks? Neural networks are computational models inspired by the biological neural networks in our brains. They are essentially a network of nodes and edges where the nodes are the neurons, and each node processes input and passes the output to the next layer. It's used in tasks like image recognition, language translation, and even medical diagnosis in our case. The the three parts of the neural network are the input layers, the hidden layers, and the output layer. Next slide. Okay, neuron model and activation functions. So if we zoom in to just one neuron in a neural network, it applies a function similar to a linear regression. So as you can see, it says z equals w1 x1 plus w2 x2 all the way till wn xn plus b. Here, W1, W2, all the way till Wn represent the weights we have in a neuron, and B represents a bias term. X1, X2 till Xn are the inputs we had received from the previous layer. Now, in the neural network, after we apply this linear transformation, we pass it through something called an activation function. Common activation functions include the sigmoid function, the ReLU or rectified linear unit functions, etc. So why do we do that? Because activation functions introduce a sense of non-linearity in our model. So when we stack multiple neurons and multiple layers with only this linear transformation, what it finally results in is just another linear transformation. So it's equivalent to just using one node of a neural network. So when we use non-linear activation functions, this allows the network to learn and represent more complex patterns and decision boundaries and move from just linear data to more complicated data. Next slide. Okay. So from a small neuron, we can now generalize to the entire network. So the neural networks make their predictions using a process called forward propagation. So as we saw in the previous slide, the output for each neuron in a neural network is calculated using this formula where sigma is the activation function, W are the weights, X are the inputs, and B is the bias. Next slide. Backpropagation and training. Okay, so now that we use forward propagation to predict and test our neural network, how does the neural network learn? This is done using backpropagation. The first step for backpropagation is to find the error between the predicted and the actual outputs. This is computed using a loss function. And the error that we have found using the loss function gets backpropagated through the network to update the weights using gradient descent. You can see the formula. This is the partial derivative of L with respect to W equals the partial derivative of L with respect to A times the partial derivative of A with respect to Z times the partial derivative of Z with respect to W. It's just a chain law. And L represents the loss function, and W represents the weights being updated. A is the activation function, and Z is the output in a single neuron. Next slide. And this is just a summary of how a neural network gets trained. The first step is initializing the weights randomly, then forward propagation to compute the output, then loss function to compute the loss, that is the error between the predicted and actual values. Then we back propagate this error to compute the gradients, and finally update the weights using gradient descent. This is repeated for multiple epochs until the model converges or the loss gets minimized. Next slide. Okay, so what we just looked at was the simplest form of a neural network, also known as the multi-layer perceptron. Now, many types of neural networks use that as their foundation, such as CNN, RNN, uh, long short time memory models, and so on. So, what a CNN is a type of deep neural network that is primarily used for image recognition. It adapts the simple structure of the MLP, the multi-layer perceptron, and is better suited for computer vision and image processing. So, it consists of three main types of layers: the convolutional layers, the pooling layers, and the fully connected layers. We can also separate this into two different tasks. One is of feature extraction done by the convolutional and pooling layers, and the other is the classification done by the fully connected and output layers. The convolutional layers extract the feature maps using kernels or filters. The pooling layer reduces the spatial dimensions of these feature maps and retains important information and excludes the noise from the feature maps. And finally, the fully connected layer performs the final classification. We can look into some of the layers in depth. Next slide. Okay, the convolutional layers. This is the core building block of CNN. Each neuron in this layer is connected to a small region of the input image. This is called the receptive field. Now the kernels or the filters slide across the input image to extract features like textures and shapes. This is done using like a sliding window technique. And the output of this operation gets converted into a feature map. And this filter is applied over the entire image, allowing the network to detect features regardless of the location. Next slide. Yeah. The second part is the pooling and fully connected layers. The pooling layers reduce the spatial dimensions of the feature map while retaining important information. There can be two types of pooling. One is max pooling and the other one is global average pooling. So max pooling, what it does is only taking the most important features and not considering the other ones. And global average pooling is when you take the average of certain features. So you're still reducing the spatial dimensions of the feature map, but you're still retaining some of the information or all of the information in the feature maps. And the fully connected layers is the final stage of the CNN, and the feature maps are flattened into a vector and passed through these layers for classification. In our case, binary classification. This is done using an activation function like sigmoid or softmax, depending on the task. Next slide. Okay, just to summarize, we have talked about neural networks, the basic neural network that is the multi-layer perceptron. We have talked about CNN and how they connect to the multi-layer perceptron and adapt for image recognition. And now we're going to talk about the ResNet 50, which is a pre-trained model on the ImageNet dataset. So the ResNet 50 is a deep neural network architecture consisting of 50 layers. It uses residual connections or skip connections to mitigate the vanishing gradient problem. What's the vanishing gradient problem? So it's when we increase the depth of a neural network, we can see that instead of the accuracy increasing, after a certain point, it starts to decrease. This is because when we perform backpropagation, the gradients while passing through the layers cannot move smoothly and sometimes they just vanish or become zero. So the core idea is that of the ResNet 50 is instead of each layer learning a full transformation, it just learns the residual or differences from the input to the output. Next slide. Okay, so why did we choose ResNet 50? Because one is that it solves the vanishing gradient problem. It's also efficient for training deep networks. It's pre-trained on large datasets and proven performance in medical imaging. We have also tested other pre-trained models like the VGG16, VGG19, GoogleNet, etc. And we found the ResNet 50 to be the best in our case. Next slide. Okay, now that we know all the different parts, let's try to put this together for our project. So the first thing is the MRI database. That is our data set. It has about 5,000 images. And then we take one single input image. We perform some pre-processing and data augmentation on that. And for each of those images, we find the histogram of gradients, the feature descriptors using histogram of gradients and local binary patterns, as well as use a simple CNN for the task. So after we have found three different feature maps, one from histogram of gradients, one from local binary patterns, and one from the CNN, we combine them into one feature vector. Uh, After that, it's passed through uh, additional dense layers, which is basically a fully connected layer, and then we perform our final classification on tumor or no tumor. Next slide. Uh, yeah, this is basically the summary of what we just talked about. We can move on. Next slide. Okay. So
We also
Aniruddh, do you like to explain the SFK?
We also implemented stratified k-fold cross-validation or SFK to ensure that our model generalizes well. We use this because, especially in medical imaging datasets, the proportion between images with tumors and non-tumors is not equal. SFK ensures that every fold has a similar proportion of tumor or no tumor images. It's trying to make a 50-50 split between tumors and no tumor images. How it works is that first we have a data set which is divided into k equally sized folds. So we use five-fold cross-validation. So for example, if our data set has 100 images, each fold has 20 images now, and there's five folds. We iterate in each iteration, one fold is used as a validation set, which is also known as a testing set because we test the model's performance and ability to classify on this set, and the remaining k minus 1 folds are used for training. So in our case, we have, for example, we have 20 images as the validation set and 80 images as a training set. And we iterate this k amount of times or 5 times. So every single time the validation set changes and the training set also changes. This allows, so then the model is trained and validated 5 times or k times, and the average performance is reported. So this allows the model to be trained and validated in equal amount of times and helps avoid overfitting because of the robust evaluation across different subsets. So this ensures that the model is being trained in even amount of times and being tested in even amount the same number of times. Next slide. Now, now that we've explained the methodology of our paper using feature descriptors and using CNN models, now let us see the results of our project.
Okay. So the four performance metrics that we used for our results were accuracy, precision, recall, and F1 score. So accuracy in our case is the number of correctly predicted, the percentage of correctly predicted images as compared to the total number of images. The precision is the predicted positives divided by the correct positives compared to the total number of predicted positives. The recall is the correctly predicted positives as compared to all the actual positives in the dataset. And the F1 score is just the harmonic mean of the precision and recall and shows how we have balanced both the metrics. So accuracy, we had a 99.73% accuracy. Precision, we had 99.88%. Recall, we had 99.56%. And our F1 score was 99.72%, which shows really high performance metrics. Next slide. Okay. So another test we performed in our project was to compare the graphs of the validation and training accuracies and losses. So in the first graph, we can see the validation loss and training loss being compared. Blue line shows the training loss and the orange line shows the validation loss. So what we can see here is some after a certain number of epochs, both the lines have converged into a single line. This shows how, as the epochs pass, our model is generalizing extremely well and not overfitting. We can see the same for the accuracy comparison, where both of them start out very far away, but as the epochs progress, they converge into one line, showing how both of them are being how the model is generalizing to the new images based on its training and not overfitting. Next slide. Okay,
Now,
Anirudh can explain
let
the
us,
confusion matrix.
now let us look at the confusion matrix. A confusion matrix is a visual representation of true negatives, true positives, false negatives, and false positives. Over here, the darkened blue areas show the true and false negatives, true negatives and true positives, showing that the majority of the model's predictions were accurate. And we have 14 images where 3 are false positives and 11 are false negatives, where the model was inaccurate in its predictions. This shows that almost out of 5,000 images that it was inaccurate 14 times, and it proves how our model is generalizing well and has superior performance across the board with all metrics. Next slide. Finally, let me show you a comparative analysis. Over here, our proposed model was compared against three different popular CNN models. The base ResNet 50 without any of the hog feature descriptors, VGG16 and AlexNet. As you can see, our proposed model demonstrated superior performance across all key metrics, where it's above 99% for every single one, while the ResNet 50, VGG16, AlexNet aren't even crossing 92 or 93% for some of the metrics. This shows that even with the same methodology, our implementation of feature descriptors made a huge impact on the accuracy and on all of the key metrics and our model's ability to classify a image whether there's based on whether there's a tumor or not. Next slide. Now let me talk about future work and developments. In our project, we did binary brain tumor classification, which means that we either detected whether there was a tumor in the image or there wasn't. What we could do to enhance our work is brain tumor segmentation, where it finds the exact location of the tumor in the image and can highlight parts which are cancerous or deadly. Secondly, we can enhance our accuracy with a reduced data set. In places where data sets and images are quite sparse, we can try to enhance accuracy with even smaller data sets using other methodologies and different feature detectors. Finally, we can use machine learning to develop a comprehensive surgical plan with the object of tumor detection. This means that essentially the machine learning model gets the image and it can lay out a precise plan on exactly where to make incisions and where to operate on the body, on the brain, and it effectively remove the deadly parts of the tumor. Yeah. Um, Next slide.
Conclusion. So, our study proposed a new ideology or methodology for brain tumor detection, offering greater accuracy, and it can also serve as a great aid for radiologists. So, the main conclusion we got from this study is sometimes when we want to make an increase in accuracy, we don't always need to increase the complexity of our models, but sometimes we can just diversify our methodology and use intelligent combinations such as feature descriptors or other attention mechanisms and etc. So this study advances AI assisted medical diagnostics and computer vision in medical imaging. Thank you.
Thank you. Any questions?
Thank you, all of you. If I will ask the audience if you have any question for Aniruddh and Ahan's project, then please either comment in YouTube or you can also email us. We will forward the questions to the students. It was a wonderful project. Thank you for joining in, Ahan and Aniruddh. Excellent work.
Thank you, sir.
Thank you.
Yeah, and of course, Shaif and Muskan who assisted and helped the kids. So fantastic work. So there is a question in the chat. So Geet Modi is asking how your work can be used for real world problems. So can anyone respond?
Yeah.
Maybe.
So we can use the same methodology and we can implement it where like every day, like doctors use this as a tool to enhance their diagnosis, maybe like as a separate way to like check that their predictions are accurate. And even with further implement iterations where we are doing brain tumor segmentation or developing a comprehensive surgical plan, it can greatly assist surgeons and doctors in identifying these tumors and removing them.
Yeah, and adding on to that, we can also develop our project to make it more computationally efficient, where uh, sometimes when we don't have the computational power, we can still make a low cost type of binary brain MRI classification, which can aid radiologists in even economically advanced and poor countries.
Right. Excellent. Thank you again for the question and the response. I will conclude the session for today, and I hope that your work continues and we do something really nice. Okay. All right. Thank you,
Thank you.
guys. Bye. I'll end the live stream now. And